important chapter in entire parasitology, Plasmodium. How many species are there? 4 species for a student who is writing a 10th exam, but you are writing entrance, PG entrance. So, there are 5 species, and Plasmodium vivax, falciparum, malariae, oval, and the last is Plasmodium knowlesi. And all these are, of course, knowlesi is not found in India yet, but it is endemic in other places. So, you have to remember something about knowlesi also. So, without wasting much time, let us now directly go to the life cycle. Malaria life cycle is again very important for exam. Definitive host, intermediate host. Which is man? Intermediate host is man. Definitive host, which mosquito? Female Anopheles mosquito. And they say that if the mode of transmission is mosquito bite, then the infective form is sporozoite, but mode of transmission can also blood transfusion and vertical transmission from mother to fetus. In these 2 cases, the infective form will be merozoites. Directly merozoites can be the infective form. Okay. And in such case, if in blood transfusion, what are the differences? Merozoites, so transfusion associated malaria, how it is different than the mosquito borne malaria? If it is transfusion associated malaria, then of course, as I told you, merozoites are the infective form, and directly the merozoites will be entering into the blood. So, there is no liver cycle. If there is no liver cycle, there is no hypnozoites. Hypnozoites are the dormant stage in liver. If there is no hypnozoite, then there is no relapse. If there is no relapse, there is no need to give the drug of choice of relapse, that is primaquine. All these are the differences between a transfusion associated malaria and a mosquito borne malaria. Okay. So, with this now let us start what happens in a normal case when the female Anopheles mosquito bites. What happens? When the female Anopheles mosquito bites, they say that the sporozoites are the infective form, they are released to the skin. From the skin they will enter to the blood, and in 30 minutes from blood they enter into the liver. In liver, 90% of the sporozoites they transform into trophozoite. The liver cycle is called as hepatic cycle or pre-erythrocytic or exoerythrocytic cycle. Both all are same, pre-erythrocytic, exoerythrocytic, hepatic cycle. And in liver, the 90% of the sporozoites they form trophozoites. Trophozoites are the feeding stage of the parasite. They will multiply. They multiply and they will grow in number. After some time they divide, and they divide by pre-erythrocytic zygogony. They undergo pre-erythrocytic zygogony to produce a pre-erythrocytic zygont. Pre-erythrocytic zygont. Pre-erythrocytic zygont undergo zygogony to release the pre-erythrocytic merozoites. To release the pre-erythrocytic merozoites. Pre-erythrocytic merozoites are the infective form to RBC. they infect RBC. So, this happens to how many cases? How much of sporozoites? 90%. The remaining 10% of sporozoites, they are as lazy as you, they will become hypnozoid inside the liver. Hypnozoids are the dormant stage of the parasite, which do not cause any infection, but 2 to 3 years later they will produce relapse of malaria. And can you tell me how relapse is different than recrudescence? Please remember, this hypnozoid is a stage is seen only in V for vivax and oval. Only vivax and oval 10% will become hypnozoid. Falciparum all will become trophozoid. There is no hypnozoid in falciparum. In contrast, recrudescence is a phenomena which is seen in falciparum and malariae. Falciparum and malariae, these two you can get recrudescence, and recrudescence is due to drug resistance. Relapse is due to hypnozoids. Recrudescence is due to drug resistance. They say that recrudescence occurs 2 to 3 weeks of treatment. Relapse occurs 2 to 3 years of primary attack, and 2 to 3 weeks after treatment, the drug resistance Plasmodium falciparum, if they are resistant Plasmodium falciparum, 2 to 3 weeks of treatment later they can cause a second episode of malaria, and this is called as recrudescence of malaria. This is seen in which species? Falciparum and malariae, whereas relapse is seen in V for vivax and oval. So, now come back to the life cycle, because we have discussed only half. Let us discuss the other part of the life cycle, RBC cycle. So, which is the infective form to RBC? Pre-erythrocytic merozoites. So, inside RBC, they say that the pre-erythrocytic merozoites inside RBC, they will grow to form trophozoites. Trophozoites, first they will form early trophozoites, that is ring form. Early trophozoites are nothing but they are the ring form. Early trophozoid later on will become late trophozoid. Late trophozoid will multiply to form zygont. Zygont undergoes zygogony. Zygogony releases the erythrocytic merozoites. Erythrocytic merozoites, once they are released, how they are released? By the bursting of the RBC. They say that RBC, the zygont, when they are released, they will burst the RBC. RBC will be lysed, so that erythrocytic merozoites will be released out. So, when they are released out, along with that, the hemoglobin digested byproducts also will be released, which are called as hemojoin. You know that inside RBC hemoglobin will be there, and hemoglobin is the nutrition for the parasite. Malaria parasite feed on hemoglobin and degrade hemoglobin to produce certain byproducts called as hemojoin pigment, and along with the merozoites, the hemojoin byproducts are released, and they are the lethal byproduct which is responsible for various clinical manifestations like fever, chill, rigor. All these clinical manifestations are due to the release of hemojoin pigments. And please remember, after the merozoites are released, these merozoites will again attack the RBC to repeat the cycle. Once the merozoites is released out, RBC cycle is over. So, they will again attack fresh RBC. They will again attack fresh RBC to repeat the cycle. Or if they are, or they will become bored of the person, they will transform into gametocyte. Gametocyte is the infective form to mosquito. Gametocyte is the infective form to female Anopheles mosquito. So please remember, in this context, the RBC cycle duration is very important for the manifestation to take place. Yes or no? At the end of the RBC cycle, the hemojoin pigments are released, hence the manifestations they appear. So the duration of RBC cycle is important for the periodicity of the manifestation. For example, Plasmodium vivax and Plasmodium falciparum and Plasmodium ovale, all these three, the periodicity is 24 hours, I am sorry 48 hours. The RBC cycle lasts for 48 hours. That is why they produce tertian malaria, that is fever occur every third day. Every third day the fever appears. For Plasmodium malariae, the fever occurs every 72 hours, that is every fourth day, which is called as quartan malaria. Plasmodium knowlesi, the periodicity is 24 hours, hence fever occurs every day, every alternate day, which is called as quotidian malaria. Quotidian malaria. Okay, so this is how the manifestations occur. So whatever I have discussed, this is about the life cycle of malaria parasite, and they say that the clinical manifestation, the benign manifestation which is common to all species, that will be fever with chill and rigor, anemia due to burst of RBC, and splenomegaly, because spleen is the site where they will try to engulf the parasitized RBCs and they will try to destroy them. As a result, the spleen will enlarge in size. So this is seen in all species, because these are the features of benign malaria. There is another form of malaria called as malignant malaria, which is seen only in falciparum, and this malignant malaria, of course, the pathogenesis I will tell you first. Why malignant malaria is seen only in falciparum species, not in other species? The reason is falciparum undergoes, I mean in falciparum various specific pathogenesis events take place which are not there in other species. One of the important event is sequestration of the parasite. Sequestration of Plasmodium falciparum by cytoadherence and rosetting. Cytoadherence and rosetting. That this means what? They say, see, this is the RBC and this is the malaria parasite inside, Plasmodium falciparum inside. They say that Plasmodium falciparum releases a protein, expresses out a protein, secretes a protein which is called as PF-EMP protein. EMP is erythrocyte membrane protein. So these proteins it will release out. So these proteins they will come out and they will be coated on the surface of RBC. They will be coated on the surface of RBC, and these erythrocyte membrane proteins are sticky in nature. Because they are sticky in nature, this results in adhesion. Adhesion to what? One side they will be adherent to vascular endothelium. Other side they may be adherent to normal RBCs also. So, that leads to rosetting. Cytoadherence means adjacent to the vascular endothelium. Rosetting is the clumping of normal RBCs to parasitized RBC. This leads to sequestration of the parasite to the vascular endothelium. So, what you can see here, this property lead to what? See, this is the blood vessels, this is the total blood vessel, in that a part of the blood vessel is occluded. So, sequestration of Plasmodium falciparum, especially in small capillaries of deep vessels, this leads to vascular occlusion, and this is the main mechanism why all these malignant manifestations they take place. Malignant manifestations, the various complications seen under malignant tertian malaria are cerebral malaria. Why cerebral malaria takes place? Because of the brain capillaries will be occluded, leads to cerebral anoxia. Okay. Other than that, black water fever, okay, algid malaria. Algid malaria is a shock like stage. Okay, so all these are the various complications seen in malignant malaria. Then complications seen in other species. Other species complications like nephrotic syndrome. Nephrotic syndrome is a complication specifically seen in Plasmodium malariae. Nephrotic syndrome. Tropical splenomegaly syndrome. Tropical splenomegaly syndrome, it is seen in mainly vivax or maybe in other species also. Tropical splenomegaly syndrome is characterized by splenomegaly and pancytopenia and hyper IgM. Three components will be there: splenomegaly, pancytopenia and hyper IgM. Okay, so all these are various manifestations. Fine. So now let us discuss about a bit of epidemiological features. Where malaria is endemic, which is the place where maximum number of malaria cases are found? Is it India or it is outside? India accounts for only 10% of total malaria burden. The main malaria burden reside in sub-Saharan Africa, which accounts for 85% of total malaria cases. India and other SEER countries, Southeast Asian region countries, both together they account only for 10% of total cases. And of course, in the world, Plasmodium vivax is the most common species. Whereas especially India is concerned, they say that the most common species Plasmodium falciparum and vivax is almost same, you can say that 50% and 49%, almost same, followed by Plasmodium malariae. Malaria is mainly in the Tumkur and Hassan district of Karnataka. And the rarest is Plasmodium ovale. And Plasmodium ovale, they say that it is very few reports are there in India. Those places are Gujarat, Odisha, Delhi, Assam. Okay, so these are the few places where Plasmodium ovale have been reported. Okay, so Gujarat and also Kolkata you can write. Gujarat, Odisha, Delhi, Assam and Kolkata, these are the few places where plasminogen oval have been reported. Very rare, but there are some cases. You can't say that it is not there.